Hello and welcome to today's lesson. In last lesson we looked at genetic engineering, and today we're going to be looking at cloning. So in your book you need to put the title cloning. By the end of this lesson you should be able to define the term clone. You should be able to explain how a tissue culture can be made to clone genetically modified plants, and you should be able to compare the process of embryo transplants with adult cell cloning. So let's get the first definition out of the way, okay? And all keywords are in green in my lessons. So the first keyword is the word clone. Now, a clone is an organism that is genetically identical to another organism, okay? So a clone is an organism that is genetically identical to another organism. Essentially, it is an exact copy of another organism, okay? So I'm going to put that it is, and I'm going to put it in red because it's important. They are an exact copy of another organism. Now, some people who are quite skeptical might be thinking, well, what's the point in making clones? What is the point in making copies of things? Now, ultimately, it's the same as when we spoke about selective breeding. You might have an organism that has a particular trait that you want, therefore you want lots of it so that you can harvest that trait. So we can put underneath here, we can say that it is useful to make many copies of an organism with a specific trait. Now, oftentimes, well, pretty much all the time, we don't clone bacteria. Bacteria divide by something called binary fission, and that happens very, very quickly. Bacteria divide every 20 minutes. So their numbers double every 20 minutes. So if I start with one bacteria, after 20 minutes, I've got 2. After 40 minutes, I've got 4. After another 40 minutes, I've got 8. And I can keep going until I've got billions of them after 24 hours. Okay? So I'm going to put here that we don't make clones of bacteria as they divide quickly by binary fission. Okay, so we don't need to make copies of bacteria. Now, one time that we might use cloning is when we've got an organism that's been genetically modified. We might genetically modify a plant cell, for example, and we might want lots of copies of it. So this is when we would use what are known as tissue cultures. So we can say, we can put the subheading, cloning genetically modified plants. Okay, or GM plants, we can call them for short. So I can put it here in brackets, GM plants. The G and the M stand for genetically modified. So we may have started with a genetically modified plant cell that we can then clone into what's known as a tissue culture. Now, this diagram from the PowerPoint shows this process in great detail. Okay? Essentially, we're starting with the modified bacteria. We're going to start with a bacteria into which we've added a modified plasmid, like we spoke about in the genetic engineering lesson. We then infect the plant cells with that bacteria. This can make a genetically modified plant cell. So because this bacteria has infected the plant cell, it's inserted this plasmid, which has made a GM plant cell. Over time, the plant cells that have been infected with the genetically modified bacteria are screened. We want to make sure which one has taken in the gene that we want. Okay? The modified cells that have taken in the gene are then grown into plantlets in what's known as a tissue culture. If we find some plants that we think have taken on the characteristic, we control them in a greenhouse and in a field to make sure they're doing what we want them to do. And then we can take cells from the parts of the plant to produce lots of identical plants, which we can then sell. So, I'm going to leave it that big in the corner. It is on the PowerPoint should you need it and if you want to use it. And we can say that, first of all, a bacteria with the desired characteristic is used to infect plant cells. So it's used to infect plant cells. We can then say that the cells are screened to find one that has been successfully infected. To find one that has successfully taken in the desired gene. So it's successfully taken in the desired gene. Okay? The cells are allowed to grow into what's known as a tissue culture. So the cells are grown into a tissue culture. Now, a tissue culture is a very important key word, and we're going to define it down here. So, a tissue culture in green tissue culture is a process where or that uses small sections of tissue small sections of tissue to clone plants. So a tissue culture is a process that uses small sections of tissue to clone plants. So once the cells are grown, we can then take parts off the plants that have the characteristic we want to make copies of lots of new plants. So we can just add here, and I'm going to put it in red so that it's clear. Red is the important points, that plants with the characteristics we want have sections removed from them to grow clones of the plant. Okay. Now essentially what we can do is we can remove a part of the plant which can then grow into new copies of that plant. So we could break off a branch and we could plant that branch that will then grow into a copy of the original plant. Sometimes we need to use what's known as cutting powder in order to encourage them to grow. It encourages the plant to create new roots. My grandfather was terrible for this. He loved his garden, never bought a single plant from where I was aware. We always used to go to the park or to garden centres, and he would break a part off of a plant he wants, take it home in his pocket, dip it in rooting powder and plant it in his garden, and it would grow. Okay? So we can say that parts of the plant can be removed and dipped in cutting powder to encourage the growth of new roots and thus a new plant. To encourage the growth of new roots and therefore plants. Okay? So a clone is an organism that is genetically identical to another organism. They are an exact copy of another organism. It's useful to make many copies of an organism with specific traits. We don't tend to make clones of bacteria as they divide quickly by binary fission, so we don't need to clone them. For example, we could use cloning to make copies of a genetically modified plant. We can use a bacteria with a desired characteristic to infect plant cells. This then creates new plant cells that have a new trait. The cells are then screened to find the ones that have the desired trait that we want, and we grow these plants so that we can take a tissue culture from them. So we grow the plants with the traits that we want. We then take parts of that plant away to make clones of that plant. This is known as a tissue culture, which is a process that uses small section of tissue to clone plants. We can also clone plants by using cutting powder. We can break a part off of a plant, such as a branch, dip it in cutting powder, plant the new part of the plant, which will then grow roots and become a new plant. So, our second one is that we need to talk about are embryo transplants. Embryo transplants, and this is something to do with cloning in animals. So we've spoken about cloning in plants. Let's think about cloning in animals. Now, as previously suggested, there are different types of cloning inside animals. One that I've mentioned is embryo transplants, and there is a second known as adult cell cloning. Okay? So an embryo transplant is our first one we're going to talk about, and it is a key term. Embryo transplant. An embryo transplant can be defined as a form of cloning where cells are taken from a developing embryo from a pregnant animal separated and grown in culture before being inserted into host mothers. Quite a long definition, isn't it, that? However, I've taken it from the dictionary, the biology dictionary. So embryo transplant is a form of cloning where cells are taken from a developing embryo from a pregnant animal, separated and grown into culture before being inserted into host mothers. So if we were to explain this, when an organism becomes pregnant, so there's my drawing of a sperm. Let's take humans, for example, and this is my drawing of an egg. When the sperm and the egg join together, first of all, they are both haploid, so they have 23 chromosomes within them. So in orange are my chromosomes. When these join together, this is known as fertilization. Okay, we have a process of fertilization taking place where we've got the first cell of the developing organism, and in the case of humans, it would have 46 chromosomes inside of it. Again, this is due to fertilization. I don't know where I'm going to write that. I'll put it there. Fertilization. Okay. Through a process of mitosis, this will then divide into 2 cells, which will then divide into 4, which will then divide into 8, and so on and so forth. Okay, we get mitosis. And what we get, let's imagine, for argument's sake, is 4 exact copies, apologies, it's my phone, 4 exact copies of the original cell. So this is the first cell of the embryo, and then the cells would divide through mitosis to form exact copies. So we've now got 4 copies of the cell with exactly the same chromosomes in them. What scientists can do is they can take out these cells and separate them. Okay? So now, rather than having one ball of cells, you would have 4 cells that are all identical to each other. They're all identical to this cell. So they've all got 46 chromosomes still inside of them, and they're exact copies of this original cell. They can then allow these cells to grow in the lab into their own ball of cells, each with 46 inside of them. They can allow them to grow into their own embryos. So each of these, that one, each of these have got 46 inside of them because they've all undergone mitosis. Okay, they're all identical. That's the point I'm trying to make, and I regret trying to write this in for every single one, but we've started, so we will finish. So we've got 46, 46, 46, and 46. Again, this is through mitosis. Okay. And then what scientists will do is they will take each of these new embryos, which are an exact copy of this original cell or this original embryo, and put them into surrogate mothers or into host mothers. So we'll end up, that's the pregnant belly, by the way, we'll end up with 4 separate mothers that are all pregnant with a clone of the original cell. Okay? So we've taken a sperm and an egg, fertilization has happened to make the first cell of the embryo, mitosis takes place to develop the embryo. The embryo is then split into separate cells, so it's split into cells. Each of these cells go through mitosis to form different embryos. The embryos are genetically identical to these original one. So we've got embryo, embryo, and embryo. And then these embryos are placed inside what are known as surrogate mothers. These are organisms that carry the clones, but the clones don't have any of their genetic material. So an embryo transplant is a form of cloning where cells are taken from a developing embryo from a pregnant animal, separated and grown in culture before being inserted into host mothers. So it might be that you want, for example, a cow that produces lots of milk. You notice that you've got a male and a female that produce lots of milk. You know the gene that allows them to produce lots of milk. You let them reproduce with each other and they make an embryo. You then take the embryo out of the mother cow that's pregnant. You look at their genes to see if they each have the gene for creating lots of milk. You would then separate them. You would allow them to develop into embryos and you would put them in surrogate mothers. So rather than just getting one baby cow that produces lots of milk, you're now getting 4. And all 4 of these babies are identical because they share the same DNA. So it's worth putting here that the embryos, or we'll put the embryos separated in this process have the same DNA. Same DNA as each other. So by our definition from earlier, they are clones. Now we're going to continue with cloning in animals, but look at a different process known as adult cell cloning. Okay? So carrying on with our animal cloning, we now need to talk about adult cell cloning. Adult cell cloning. Okay? Now, adult cell cloning is defined as a form of therapeutic cloning, and I'm going to put it in a different color and I'll explain why in a moment. Therapeutic cloning, I can never spell therapeutic. Therapeutic cloning, where the embryo created is implanted into a female animal Okay, and I'm just going to leave it there. An example of adult cell cloning would be Dolly the Sheep. Okay, you may be a bit too young now to know who Dolly the Sheep is, but we're going to talk about her today. So we can put e.g. Dolly the Sheep. Now the reason I've put therapeutic cloning in a different color is because we covered therapeutic cloning when we looked at stem cells. If you are not sure about what therapeutic cloning is, you need to revisit that topic in your own time. So we can say that therapeutic cloning was covered in the topic on stem cells. Okay, so that was way back at the start of the course. Now, to help us understand adult cell cloning, I've got this diagram ready for us. Essentially, what happens in adult stem cell cloning, or adult cell cloning either, is we get an organism and we remove an egg cell from it and remove the nucleus. Now, we can also take a cell from the organism we want. So this is the organism that has the characteristic that we want. So I'm going to put in red that this is the organism with the characteristic we want. So for example, it might be a sheep that produces lots of fur, or lots of wool even. From the organism we want, we remove just a normal body cell, so such as a skin cell. We remove the nucleus from the body cell and throw away the rest of the cell. The reason we want the nucleus is because that contains the genetic material of the trait we want. So we can say that this contains the genetic material of the trait we want. Okay? So this nucleus contains the trait we want, whether that's having lots of wool, producing lots of milk, anything. This nucleus is removed, and this nucleus is removed because it doesn't contain any of the DNA we want. So I'm going to circle this one and say that this is thrown away as it does not contain the DNA we want, or the genetic material we want. Now this nucleus is literally thrown in the bin. We don't want it, okay? We take the nucleus containing the DNA that we want, and we take the empty egg cell from a random sheep, and we put the nucleus into that egg cell, okay? Now remember, this has the genetic material of this organism. It has nothing from this organism, so it will only look like this one. The empty egg cell that's had the nucleus inserted into it is given an electric shock. This stimulates the egg cell to start dividing. By giving it an electric shock, it causes mitosis. So we can say that the electric shock causes mitosis to take place. Once it goes through mitosis, the cells develop into an embryo, a ball of cells, that's implanted into another female, and then that female will give birth to an organism that has the exact genetic material of this one. Remember, this is very different from fertilization. Fertilization is where you get half the DNA from the father, half the DNA from the mother. However, in this process, all of the DNA is coming from one organism. So we're making clones of this organism here. So if you look at the diagram, this organism here that's given its DNA through the nucleus is identical to the offspring that's been produced. The offspring is nothing like the surrogate mother. Okay? So I'm just going to put over here that the offspring, in fact, no, I'm not going to use the word offspring, I'm going to use the word clone. The clone produced has none of the DNA or the genetic material of the surrogate mother. A surrogate mother is an organism that carries offspring that aren't their own. They carry offspring for another organism. Now, we've said that Dolly the Sheep is an example of this. The first time this was done was in, have I got a date written down? So it took place in about 1992. Yeah, 92, 91, 92. Now, Dolly the Sheep was the first sheep that was cloned this way. They got one sheep, they removed the nucleus from that sheep, they took an egg cell from another sheep, they put the nucleus from this sheep into an empty egg cell, gave it an electric shock, it developed into embryos, they put it into a surrogate mother, and the offspring that was born would have been Dolly the Sheep, and Dolly the Sheep would have been identical to this one. Now, Dolly lived until she was 6.5 years old, and she even had offspring of her own, and it showed that this technique was possible. So adult cell cloning is a form of therapeutic cloning where the embryo created is implanted into a female animal. Essentially, we take an egg cell from a female of that species, we remove a nucleus from the egg cell. At the same time, we take a body cell from the organism we want to clone and we remove its nucleus. We put the nucleus from the organism we want to clone into the empty egg cell from the other one. We give it an electric shock, which then creates an embryo, or the cells divide by mitosis to form an embryo. That should say mitosis, not mitosis. Okay? They divide by mitosis to form an embryo. We then take the embryo and implant it into a surrogate mother who will carry the embryo until it develops into a baby. Then when the surrogate mother gives birth to the baby, it will be identical to the organism from which we use the nucleus. The offspring has absolutely no genetic relation to the surrogate mother. It will only be genetically identical to whichever nucleus we used. So, you should now be able to tell me what the word clone means. You should be able to explain how a tissue culture can be made to clone genetically modified plants, and you should be able to compare the process of embryo transplants with adult cell cloning. If you have any questions at all about this lesson, do not hesitate to get in contact with me by the usual means. If not, I will see you in the next lesson. Thank you very much.